Hi, this is Miss Litton, and this is my wonderful zero-period honors bio class. Say hi.
Hello.
Cade, your bio buddy misses you. It's a little bit of a hard day for him today. You have anything else you want? Did you want to tell him something?
I just want to say I love you.
All right. So we're going to talk about how to build a chromosome. And a chromosome is built out of DNA. And you can see right here we have a DNA double helix up at the top. And what happens when you want to build a chromosome is that DNA will wind itself on proteins that are called histone proteins that become a group of 8 histones called a nucleosome. So they'll wind up on that. So we're backing away. This blue string you see on here, that's the DNA double helix. And then that winds all together. And then this twists on each other and twists and twists and twists and twists and twists until you have a chromosome. What would be similar is if I was going to take somebody who had a lot of hair and I took only 3 pieces of her hair, literally 3 hair strands, and I braided those together, and I did that all over her head, just 3 hairs at a time. And then I braided those together, and then I braided those together, and then I braided those together until I had a super braid. Okay? And that would be similar to how a chromosome forms. Okay? And I want to show you a video about that.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes.
Now they're going through the stages of mitosis right now. So that would be prophase.
It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
Okay, now you have already studied the cell cycle. How long would that normally take? Yeah, that could take about 24 hours for that. So on your notes, chromosome structure, following S and G2 stages of interphase, cells prepare for mitosis. Chromatin, DNA, and protein start to coil up into rod shapes. Chromosomes appear as double. Chromosomes appear as double. And they're called sister chromatids, 2 copies due to the S stage. They're joined by a centromere. Organization, DNA winds around histone protein spools. Histone is spelled Histone. Histone protein spools and forms a nucleosome. The nucleosome then begins coiling until condensed into a chromosome. Okay, so the DNA would appear like this, and then it'll start to coil up, but when it coils up, when it's first coiling, it's always going to coil up as a doubled 2 sister chromatids because it's already done the boom boom. Yes?
Was that last part in that video in animation, like the pink one where it showed the cell division? That was also an animation.
Yeah, that was showing you mitosis. So it was showing you the chromosomes become visible And that's going to happen at the beginning of mitosis, and then it's going to go through each of the stages, prophase, metaphase, anaphase, and telophase to form 2 cells. Okay. Read the slide.
Noted. Yeah. You guys have a bad day. It's a megabyte.
Yeah. This right here is this, right? This is this. Okay. All right. Now, you've heard of diploid and haploid. All of your body cells, your somatic cells, are diploid. They have 2 sets of information. One set of information came from your mother, and one came from your father. Okay. In your gametes, okay, in your sperm or your egg, that is not diploid. It is going to be what? Haploid. Because a haploid and a haploid, a sperm and an egg, can fuse to form a diploid zygote. Good. So when you're referring to a diploid number, okay, these are chromosomes that are coming from 2 parents. So you might think, well, our diploid number is pretty cool. We've got 46 chromosomes, but look right here, a potato has 48. Okay, so it's how you use them. Okay, so on your numbers, a diploid number is 2n when you have 2 full sets of chromosomes. One set was received from each parent, and this is normal for somatic cells. A haploid number is referred to as n. What would our n number be? 23. Because 2n, 2 times 23 is 46. Good. So our haploid number n, when you have only 1 set of chromosomes, it could be a mix of parental chromosomes to generate a sperm or an egg, and those are called gametes. And if you want to add after that, gametes are formed by what process, though? I'm bringing up diploid and haploid, but gametes are formed by what process? Meiosis. M-E-I-O-S-I-S. So you might want to put after gametes formed by meiosis. Okay, and mitosis by somatic cells is for growth and repair, but meristematic tissue in plants can always do mitosis. Can always do mitosis. That was Cade when he was a little boy.
Yeah. he was such an animal.
All right. All right. So now, this is the part where if licorice were chromosomes, this is the part we're preparing for. We need to understand the stages of mitosis. The first thing I want you to see, because this will be similar to your cells you're going to model, how many chromosomes do you see in that cell? 4. Okay. What's going to happen to them during the S stage of interphase? It's double. They're going to boom, boom, right? They're going to double. And so they will appear like this as sister chromatids. And then it's the job, when you do mitosis, to make sure a big red gets in each cell, a big blue gets in each cell, a little red and a little blue gets in each cell. So when we talk about the stages, that's just thrown on names to explain, like, as the cell divides, if we stop it here, we call it prophase. If we stop it here, we call it metaphase. Because the chromosomes are always in these key positions. So, for instance, here, in early prophase that you're looking at here, you still have your nuclear envelope. You can see the chromosomes starting to form. These right here, they're like churros. What do we call them? Centrioles. Okay, so what happens is the nuclear envelope is going to start to break down, because we need to take these doubled chromosomes and put some in this cell and some in this cell, and they're normally encased within the nuclear envelope. So we need to make the nuclear envelope go bye-bye. Okay, and then we're going to put these centrioles are going to start to become visible. And what they do is the centrioles move during prophase to either side of the cell, to either pole, till you're here, there's a set of centrioles here, And these are spindle fibers coming across. Remember when we talked about the cytoskeleton and there were microtubules? Okay, dig in right here, make sure everybody's eyes are right here so you make sure you understand it. Okay, these microtubules are going to help push and pull the chromosomes apart. And what you can think of is kind of an old school clothesline. Okay, and the centrioles are the poles, and then the strings that go along the clothesline, those are like the microtubules, and then you hang your genes on them. All right. Okay, you're hanging your genes on them, and then what you're going to do is basically rip your genes apart so one goes to each cell. So this part in here, prophase, there's a lot that's going on. Okay, the nuclear envelope, and then a nucleolus in here disappears. The chromosomes become visible, and then you have these microtubules that are forming. If we continue on, the chromosomes, you don't want to make a mistake on this, so you line all the chromosomes up, single file in the middle, and that way you can make sure big red goes here, big red goes here, big blue goes here, big blue goes here. So they meet in the middle at metaphase, right here, and then what you're seeing right here is if these were the sister chromatids, they're getting pulled apart like this, kind of getting dragged apart by their centromere, and they go away at anaphase. And then you're forming around these and around these a nuclear envelope here and a nuclear envelope here, and you can re-see the nucleolus in the middle. And then this part right here is division of the cytoplasm, and that's called cytokinesis. And those are your different stages. Okay, let's write them down, and then I have a little song to teach you. Okay, go to your notes. Mitosis generates two identical cells. Prophase, chromosomes consisting of sister chromatids become visible. The nuclear envelope and the nucleolus break down. Okay, centrosomes. Let me tell you what centrosomes are. Centrosomes are these regions where the centrioles sit. Okay, it's called the centrosomes. Okay, centrioles sit in a centrosome. Centrosomes with spindle fibers move to the poles, P-O-L-E-S, poles, and asters form. When you hear the word asters, can you think of something in the night sky? Asteroids, okay? So that's where the name comes from. It's because these spindle fibers are going across, all coming out of the centrosome. And so that's an aster. Okay? So, asters form. Metaphase, the chromosomes meet in the middle. Anaphase, the sister chromatids are pushed and pulled apart. Telophase, the nuclear membrane with a nucleolus reform. And sister chromatids, now called chromosomes, unwind. Cytokinesis is division of the cell forming two cells. In animal cells, a cleavage furrow indents the plasma membrane. In plant cells, the cell walls do not permit furrowing. Instead, a cell plate forms, which will become a cell wall. And I'll show you that. Yes?
Can you repeat the notes for anaphase and telophase?
Yeah. Mm-hmm. So for anaphase, sister chromatids are pushed or pulled apart. And then telophase, your word is reform. So here you're looking at cytokinesis. So this is everything except the nucleus. And here you're looking at plants. I would like you to ping pong back and forth. Blue, you go first. Explain what's happening in each one of these stages. It goes prophase, metaphase, anaphase, telophase. Go ahead, describe. Okay, come back to me. Repeat. The whole thing's called mitosis. Go ahead. The whole thing's called mitosis. First stage.
First stage.
Prophase.
Prophase.
Do that again. First stage.
First stage.
Prophase.
Prophase.
The chromosomes. These are like chromosomes. Okay, and these are sister what? The chromosomes become visible. See, I'm like coming up. The nuclear envelope. And the nucleolus. Disappear. Radiating microtubules. Casters. They meet in the middle at metaphase. Away at anaphase. Do it again. Away at anaphase. To nuclei at telophase. Cytokinesis. So we're dividing the cells. Okay, you got the whole thing? The whole thing's called mitosis. The first stage is prophase. The chromosomes become visible. The nuclear envelope and the nucleolus disappear, radiating microtubules. They meet in the middle at metaphase, away at anaphase, to nuclei at telophase. Perfect. Here, you got it down? All right.
Can you record it now?
Record that song.
Yeah, for your YouTube channel.
You're not ready yet?
What else do you guys
No.
need? Oh yeah, let's record this.
Okay. Okay. Who's gonna record it?
I'll do it.
Somebody gonna come up here and do it with me? Anybody else gonna come up here and do it with me? Tell her to come. Can you come do it with me?
You can
Okay.
share it.
Okay, here we go. You ready? Tell us when.
No, if I'm
All right, the whole thing's called mitosis. The first stage is prophase. The chromosomes become visible. The nuclear envelope and the nucleolus disappear, radiating microtubules. Casters. They meet in the middle at metaphase, away at anaphase, to nuclei at telophase. Cytokinesis. Yay! Yes.
So why didn't you join together at the end for cytokinesis if it's splitting apart?
Oh, I was pinching I was pinching the cell, like dividing the cell.
Okay.
I was like coming between them, dividing them.
I felt like that person that does like the sign language next to the president.
You did?
Yeah.
Was I the president?
Yeah, you were the president.
Okay, sweet.
Yeah, it was
Let's go, Mac Miller, DMX, Usher. Too slow. Yay! Your first Hyundai on the year! Yay! Come on.
We took off a while ago.
Good job, kid. Cuddy.
These are proteins that are being sent. This should be another honey. No, it's good. You've had honeys all along. You're right. You're right. I was right.
Cat and a face. Oh. Every time it's go away.
It's not going to be handy.
What? It's not?
Why is it? Did you get it wrong?
Do we have some losers? Usher! Damn it.
Yeah.
You made it sound Jewish.
You sure?
16% of you picked interface S. What happens during S? DNA is synthesized. You're not making proteins. When are you making proteins? Question. Look at your BioBuddies clicker. If they don't show you, just start ringing the shame bell. Are you shaming yourself?
I read DNA proteins. I read it wrong.
Well, you know, I'm just gonna make sure. Thank you.
I'm like, why,
Okay, somebody picked F. The sister chromatids are separating. What do we remember for that one? Away at anaphase. Somebody picked H, telophase, to nuclei at telophase. That's when you're rebuilding the nuclear envelope. Rebuilding nuclear envelope in the nucleolus epitope here.
Oh.
You guys are posted. You look actually really good. Because you can see all of your arms in the background doing the right thing. It's super cool. You're doing well.
Can you put in like the description of the video, like for cases?
Oh, one loserdom. They put telophase. It's two nuclei telophase. Division of the cytoplasm is cytokinesis. Make them show you your clicker right now, because somebody's getting a shame bell right now. Somebody's getting a shame bell.
Oh. It's a new bug. What stage? No, they say we form that space in the middle.
Let's go, commit.
Oh, cut off.
Okay, look at me. me. Chromosomes become visible, the nuclear envelope, nucleolus break down. When did that happen? Prophase. Check your bio buddies clicker because a whole bunch of you said telophase.
It's prophase. I meant prophase.
Remember, you're gonna have a quiz over this, so let's celebrate intelligence, not ignorance. They both start with I. One's better than the other.
One's more enjoyable.
Oh, that's right. I remember. Come on, it's been a long time. I've been really good.
1 week.
No, that's how long I've gone.
I feel like
No, 1 week till
I know.
inauguration day.
D-Day. Oh my gosh!
oh my god.
Drake, where is Drake?
That's me.
Drake.
Drake.
Oh man.
Oh, I'm just
Okay, look at the picture.
That's what they all say.
You studying the picture? Study. Okay.
I put it in. Oh, I already go. Give me some dough.
Three, two, one.
We're like
What? Yeah, so that photo that you saw, let me bring it back here. So they're going away at anaphase. Shane bells should be ringing.
That's not fair. That's not fair.
I don't want to play you.
That's so unfair.
Kanye, Chance, let's go. Commit. It's taking too long. Okay, stop. Stop for a minute. Should you be missing this one?
No.
How many times have we discussed it? Yeah, 6 times. I'm gonna go with 6. So DNA is replicated. What's the word when DNA is replicated? What does it start with? Synthesis. S. Okay, that would be E. Somebody still picked G2. Who is that? Let's find out. Let's find out. ASAP. Focus on the second part of it, not the first part of that question. On the nuclear envelope reforming. Focus on that.
No, I asked you if it was, and you said it actually is. And I was like, oh, okay.
2 nuclei at telophase. Guys, are you focused in? Because some of you are still missing it. 2 nuclei at telophase. All right. And then binary fission, cell division in prokaryotes, a single circular chromosome located in the nucleoid region. A single circular chromosome located in the nucleoid region. Binary fission, chromosome replicates first, then cell elongates and builds a cell membrane and wall. A cell membrane and wall between the 2 identical chromosomes. The cell membrane and wall between the two identical chromosomes. Right? And then remind me, I'm going to try to write a note. I need to show you 2 videos I accidentally skipped over.
Yes.
I need to re-show you the 2 videos. Okay, last 2 videos. All right, let's pack it up. We'll pick up meiosis next class.
I mean, my
Meiosis.